Good evening. Is it on? Good evening. On behalf of the Indian Academy of Sciences, I'm delighted to extend a very warm welcome to all of you in the hall here, and also those of you who have joined online over YouTube for this public lecture today, co-hosted with the Indian Institute of Science. The speaker today is Professor Varmus, who is visiting India presently as a Raman Chair Professor of the Indian Academy of Sciences. And instrumental to several aspects of his visit to India this time has been Professor Partha Majumdar, the immediate past president of the Indian Academy of Sciences. And I'm pleased to invite him to introduce the speaker and conduct this session. Please, Partha, over to you.
Welcome everyone. This is indeed a great moment for us. We have Professor Harold Varmus with us this evening. He is the Raman Chair Professor of the Indian Academy of Sciences. As many of you may know, the Raman Chair Professorship was instituted by the Indian Academy of Sciences in 1972. Professor Harold Varmus was selected as the Raman Chair Professor in 2020. However, he wasn't able to come to India for obvious reasons, and he was very kind that he's here today with us. He's not only here today, but he is actually visiting several institutions in several cities of India. I'm going to tell you very briefly about his life, his career in science, his major contributions, but of course to narrate his contributions will take a very long time, so I'm going to be short. When Dr. Varmus was young, he found himself drawn to the humanities. He immersed himself in Shakespeare, Dickens, and poetry. And I quote from him, "I wasn't getting the same level of inspiration from science. My impression of science was that it was something that was more predictable. I didn't see the adventure in it. I didn't see the imagination in it." However, years later, he had the following to say about science, and I quote, "It's a drug, it's an addiction, it's a craving, I have to have it." Following his father's footsteps, he did his MD in 1966 from Columbia University's College of Physicians and Surgeons. He was fervently opposed to the Vietnam War and didn't want to be drafted, so he joined the Public Health Service at the US National Institutes of Health in 1968. In 1970, he moved to the University of California at San Francisco and joined J. Michael Bishop's laboratory. He was to win the Nobel Prize in 1989 with Michael Bishop. The research of Bishop and Varmus produced a string of findings that changed the approach to the study of cancer. They discovered that oncogenes that cause cancer are necessary parts of a cell's genetic structure that have accumulated some random changes during cell division or by exposure to adverse environmental agents such as radiation or tobacco smoke. They made their discovery by studying a group of RNA viruses called retroviruses, some members of which are strongly associated with cancer. Varmus and Bishop were awarded the Nobel Prize in Physiology or Medicine for their discovery of cellular origin of retroviral oncogenes. After leaving NIH in 1970, he returned in 1993 on invitation of President Clinton as the 14th director of the US National Institutes of Health. Dr. Varmus was one of the most successful directors of the NIH. During his tenure, he was able to make some stellar appointments to the leading positions of many institutes within the NIH system, make remarkable improvements to the scientific infrastructure of NIH, double its budget, and initiate many training and global outreach programs. Dr. Varmus has spent nearly 5 decades in cancer research. He has contributed strongly to our understanding and potential treatment of cancer. Dr. Varmus has predicted long ago that immunotherapy will be of much wider use in cancer, and I quoted that from one of his writings. We now know how prophetic that statement was. He left NIH directorship in 1990 and became the president and CEO of the Memorial Sloan Kettering Cancer Center in New York. On being called upon by President Barack Obama, Varmus assumed the position of director of National Cancer Institute in 2010. He resigned from NCI in 2015 to return to New York as Lewis Thomas University Professor of Medicine at Weill Cornell Medicine. Dr. Varmus visited India first when he was a medical student and quite regularly in the last several years. He has provided advice to the establishment of a major cancer center in India and encouraged scientists to undertake meaningful research on cancer genetics and cancer biology. Dr. Varmus is an ardent believer and promoter of open access to science. He helped launch eBiomed, which later became PubMed Central. He is a co-founder of the Public Library of Science. I would like to add that his erudition in all aspects of life, including arts, literature, science, society, seems unbounded, and anyone who has spent a few hours with him will vouch that. And his humility will put each one of us to shame. It's really a joy to be with him. I present to you Dr. Harold Varmus, who will speak on Axioms for a Career in Science. Dr. Varmus. Yes, you do.
Well, it's very good to be back in Bangalore. Been here a couple of times before. The last time I was here, about 20 years ago, I was here with my younger son, who so. Okay, this shouldn't be so complicated, but now I can hear that I'm much more audible. So, I was simply reflecting on the fact that it's nice to be back on this campus where I've been at least twice before. I'm very grateful to the Indian Academy of Sciences for selecting me to give these lectures around the subcontinent, and particularly grateful to Partha, who has stuck with me and taken on the burden of arranging my complicated and exhausting itinerary. And I'm grateful to him for that elegant introduction and the summary of my career that will probably help link together some of the things I will talk about today. So my goal here was to tell you about some aspects of my life in science. I have to recognize that life is not infinite and that there comes a time when you have to think about what's happened and what other people should learn from your own career. And I'm going to try to do that not by being overtly historical, but instead by telling you about some of the principles by which I've tried to conduct my life, and some of the principles that I think you might want to consider for yourselves. And I will do that by illustrating how applications of these principles have affected my activities in various aspects of science, ranging from actual research to teaching and to political activities. First, let me tell you what an axiom is, at least in my view. The Cambridge Dictionary says that axioms are statements or principles that are generally accepted to be true. Maybe they're not, but nevertheless we live by them. And what I'm going to be telling you about is a set of axioms, 10, that have worked for me. They may not work for you. And I think the point that I would like to emphasize in the beginning is that I'm not giving you a prescription. I'm just telling you what has been meaningful in my own life, and you have to choose yourselves how you're going to be guided in your own activities by your beliefs about what your work should mean. The first aspect that I want to reflect on is something that Partha actually alluded to, which was my prolonged intellectual adolescence. That is, my inability to come to some conclusion about whether I should spend a life as a physician, as a scientist, as a student of the humanities. I need to do this by introducing a little bit of autobiographical information. Partha very kindly stressed my humility. I'm not too sure that too many people who know me well would think that is my primary attribute, and here I'm going to seem probably less than humble. I also want to emphasize that, yes, a prolonged adolescence is a nice idea, and I don't think we should be shoved into a career path at the age of 16 or 17, as some educational systems do. But nevertheless, once you make up your mind, I would argue, seize the day, because our youthful energy doesn't last forever, and when you have it, it's the time to do your most important work. So I was brought up by people who already were professionals, even though their parents escaped persecution as Jews in Eastern Europe in the end of the 19th, the early part of the 20th centuries. And one of the things that keeps me very fond of my own country is that America has been a place where people can arrive after decades of suffering at the hands of persecution and rise quite quickly. My grandparents never went to college, and yet their children, born just after my grandparents arrived as immigrants, went to Ivy League schools, Harvard and Wellesley, and on to professional training. My mother to become a psychiatric social worker, my father as a physician, a family doctor. When I think back on this history, I'm always conscious of the kind of privilege that I've had that does make it easier to have the kind of career that I've had. And if anything makes me humble, it's the sense that my upbringing was made a lot easier by the environment into which my grandparents moved. After studying in public high school, a very ordinary high school with only a fairly small fraction of students going on to college, I was fortunate to end up at a small college, Amherst College in Massachusetts, where my initial intention of being a biology major and a pre-med, heading off to medical school as might be expected of the son of a Jewish physician. I became seduced by literature, did a thesis on Charles Dickens. Then, rather than go to medical school, which had been my initial plan, I went off to graduate school in English literature at Harvard for a year or so. Was somewhat dissatisfied by that experience and decided, after all, to go to medical school at Columbia. Had a good time there, as you heard, spent some months in Bareilly, UP, learning something about the status of healthcare in India, and then came back to America, did a couple of years of training in internal medicine. At that point, the Vietnam War obliged all male medical school graduates to do some kind of national service or spend time in jail or in Canada. And fortunately, one alternative was to work in the public health service, and one component of the public health service was the National Institutes of Health, where I and many of my male colleagues who were fortunate enough to be selected into training programs went to learn science. And indeed, I was 28 before I did any really serious scientific work. At the time that I went to the NIH, I realized that science was exciting. And Partha has given you some quotations, some of which are pretty obscure, and I'm impressed by his scholarly energy in digging these up. But the fact is, as I'll mention to you in a moment and try to illustrate, the science that I was able to do at that period was quite thrilling, and it changed my mind and made me decide that I wasn't going to practice medicine. I wasn't going back to a life in literature, that I was going to do science, and I tried to move quickly to find something that I was enthusiastic about studying. So, once this happened, I tried to make a rapid end to my adolescence and move as quickly as possible to a situation in which I could be an active investigator. And I think it's useful in the current context of the conduct of science, of the conduct of a scientific career in America, to contrast what happened when I made up my mind. That I was 28 when I went to NIH as a member of the Public Health Service and learned to love doing science. I knew, as you'll hear in a moment, that I wanted to change the kind of work I was doing from studies that I'll describe in a moment of gene regulation in E. coli to work on something that was more medical in character, but allowed an approach that was that proceeded through fundamental science. And so I went to UCSF initially as a postdoctoral fellow, but quite quickly was able to get an appointment to teach medical students and become an assistant professor, and was able to obtain grants because grants were more readily available then than now. And by the time I was 38, I was ready to do a sabbatical in England. I'll say more about that in a moment as well. But now for people aspiring to do science in America, life looks quite different, and careers develop more slowly. And one might decide to become a doc at the age of 23 or so, spend 7 or 8 years getting appropriate degrees, MDs, PhDs, or both, be a postdoctoral fellow in 1 or 2 labs, get a first job at the age of 38 or so, maybe the first grant 4 years later, and then if you're lucky, take a sabbatical when you're really ready for retirement. So these are, they're amusing comparisons, but many of us who are senior scientists are concerned about what's happened to the pace of development of careers because of an atmosphere that we consider to be hyper-competitive. Not enough jobs, not enough grants, not enough slots in prestigious journals, defective tools for evaluation of careers, creating an atmosphere for doing science, especially biomedical science, that is hazardous to one's mental health. And my colleagues, Bruce Alberts, who has been here in the past, Mark Kirshner, Shirley Tillman, and I wrote a piece about 8 years ago, summarizing some of the flaws that have resulted in this delayed chronology and the tensions that currently exist in medicine. And we need to confront those ideas and begin to think about solutions to problems that most of which I won't have time to talk about today, but I urge those of you who think about the characteristics of life in science and the demography of science and the dynamic between choosing to do a career in science and the requirements of having the right kinds of jobs and the right kind of support, because all of us need to pay attention to the environment in which we work. So the second axiom I'd like to say something about is a different aspect of the environment. I've found that one of the most important things to do is to be around people who are smarter than you are. And I can't stress enough the importance of feeling that you're with people from whom you're consistently learning. I'm not going to spend a lot of time talking about that. It becomes sort of self-evident once you begin to think about who you want to spend your time with. But just to give you some illustrations of the kind of people I mean, this is a picture that shows my mentor when I was a trainee at the NIH, Ira Passon, who's still an active scientist at the age of 91 at the NIH. My good friend David Baltimore, who was one of the leaders in virology when I elected to move into the study of cancer viruses just after I left the NIH. But the most important person here is Mike Bishop, who I know has visited India several times in the past and became my laboratory partner for 15 years or so at University of California, San Francisco. And Mike has written in his own memoir about our relationship. You can read that for yourself, but the idea is that a feeling of mutual respect and a willingness to share ideas and work habits and social events and a lot of other things makes life as a scientist, in my view, much more pleasurable than it might otherwise be. There is a tendency in the US and possibly here as well to worry about people's independence. To my view, the important thing is what makes an environment in which you can produce good work, and often that means working closely with one or two or three other people and guide together a team of trainees who produce great work. And whether you can tell or whether there's one person driving the work or not is less important than the pleasures and the productivity of the team effort. The third theme has to do with what is really critical in a scientist's life, that is, choosing scientific questions. Now, all too often I find that my trainees are thinking about what would be a good topic to work on to get grant support, or what might be appealing to prestigious journals, or what might be a way to satisfy some aspect of the current environment. What are people working on that looks productive? And I think that there are some clear criteria for choosing a scientific question to pursue, and those include believing that the question is really interesting, that the question addresses problems that are long range, that is, they're going to give rise to answers which produce additional questions, and those questions will produce answers that generate yet more questions. And finally, that the kind of questions that you're asking can be answered with technologies that are either available already or very likely to become available in the near future. And it's also worth thinking about whether the questions that you're asking might at some point have societal benefit, which for most of us who are in biomedical science means might be applicable to maintenance of health and treatment or prevention of disease. And sometimes the questions that one tries to ask are not simply chosen by you, but instead thrust upon you. And that was the case, of course, that I exhibited when I was starting off at NIH and really didn't know much about science when I joined Ira Paston's lab. And Paston and his colleague Bob Perlman had made an observation in the late 60s that helped to solve the problem of dioxy, which is a problem of cell metabolism discovered by Jacques Monod in the 1950s, when he observed that when E. coli were grown in glucose, they didn't make a product of the lac operon called beta-galactosidase. And it wasn't clear what was going on or how this phenomenon was operating, but a suggestion had been made based on the work of Earl Sutherland, who won a Nobel Prize for discovering cyclic AMP. Paston thought, well, maybe cyclic AMP is not just an important messenger in mammalian cells, but also a regulator of gene expression in E. coli, and that would reduce the problem of understanding how certain hormones regulate gene expression in mammalian cells by studying cyclic AMP's ability to regulate genes in bacteria. So the question is whether, what Paston and Perlman had shown was that when you add an analog of cyclic AMP to bacteria growing in glucose, you can overcome glucose repression of the induction of beta-galactosidase. And the question was then, how does this happen? This is a result of an effect on transcription of the lac operon, translation of lac messenger RNA, or on the stabilization of lac RNA or protein. And the problem was, how do you measure the regulation of a single gene, especially in an era in which there was no molecular cloning to deliver an isolated gene to you, and you had to use some tricks to measure gene expression at the transcriptional and translational levels. The technical solution, molecular hybridization, I won't belabor that. I'm sure everyone in the audience is familiar with that. And we were able to develop an assay based on bacteriophage that carried the lac operon and the use of deletion mutants of E. coli that lack the lac operon, and make an assay very clear. This is the kind of result I like to report, where the gene being on, it gives you a steep upward curve, and the gene being off gives you a flat horizontal line, and the answer is really, really clear. There's some tricks that were used, I've just mentioned them, but basically you have an assay, you suddenly are powerful, and being powerful is a joyful moment. And then you can do the actual experiment of growing cells in the absence of inducer, in the presence of inducer, where lac expression, lac RNA levels are high. You add glucose and levels are low, that's glucose repression, and then you add back a cyclic AMP analog, and suddenly the levels of synthesis of lac RNA are rise dramatically. This gives pleasure. This gives you an opportunity to tell your neighbors that, humbly or not, that you've solved something. And indeed, you know the answer. Cyclic AMP increases the transcription of the lac operon. That's a result. But it's a result that just prompts more questions that others came to answer, namely, how does that happen? What does cyclic AMP do? Does cyclic AMP have a binding protein that contributes to the transcriptional apparatus to make transcription operate? And so, like all good scientific questions, this question I was presented with led to answers that prompted yet more and actually deeper questions. So I learned a lot from these 2 years at the NIH, and I learned that having a simple assay that gives you a clear answer is a great thing. Maybe it's not sex, but it's almost as good. Secondly, that simple models are fun to work with. We were working with cyclic AMP and the lac operon not because we cared about bacteria, but because Ira's lab was interested in hormonal control that we knew involved cyclic AMP as a second messenger. Third, as I've tried to emphasize, a clear answer leads to more questions. Some of those questions may be fairly mundane, but some of them, like how does cyclic AMP actually work in a cell to turn on the transcriptional program? Those are profound. Then I also learned that one of the joys of science is not necessarily just struggling at the laboratory bench, but also telling your peers about what you've just done. And as others have commented, famously Francis Crick, the gossip factor is really important. If you enjoy telling somebody about the work you've done, you know that you've found something to work on that you really care about. And finally, for someone like me who had the perspective of an MD who had done some clinical training, I learned that these methods that were just then evolving of molecular hybridization and other things that actually have proven to be indispensable in developing our modern view of how cells work, that those things could be applied not just to simple organisms like E. coli, but also to complex human diseases like cancer. So I was ready to move on to something else and to seek a whole other set of questions, and I was ready to move. I wanted to move to California for reasons that had nothing to do with science. It had to do with my interest in carrying a pack into the wilderness and go trout fishing and live a mountainous life on the weekends. But in trying to choose what to do next, in addition to thinking where I wanted to live, I had been influenced by the courses that were then available at the NIH and by my fairly widespread reading to recognize that among the diseases that seemed ready for more intense study was the set of diseases we call cancer, and that there were some particularly attractive ways to get into the problem. Remember, this is before molecular cloning, before sequencing, before you can do all these things that students today are familiar with as some natural, not natural phenomenon. They weren't. They had to be created. But at that time, the one thing that allowed you to concentrate on a few genes rather than thousands of genes and thinking about the potential genetic origins of cancer were viruses found in many animals that we call tumor viruses. Some of them carried their genes as DNA, some as RNA. For reasons that I won't have time to discuss, I made a choice that to work with the viruses that carry their genes in the form of RNA, then called RNA tumor viruses, now called retroviruses, because the RNA genomes of these viruses get turned into DNA by reverse transcriptase, something that I suspect everyone in this room knows. As I thought about the problem, I thought there were two big questions that I'd like to try to address over the next several years. First, how do these viruses grow? And there was already an hypothesis out there, first promoted by Howard Temin, called the provirus hypothesis, that claimed that somehow the RNA of the genomes of these viruses was turned into DNA by an enzyme that had not yet been discovered, and that the DNA was linked to the host chromosome to be perpetuated in cells that had become cancerous as a result of the infection. So how does that all happen? What are the enzymes? What are the conversions of the RNA genome to DNA? What are the means by which the DNA form of the genome, the provirus, is transcribed to make both progeny RNA and messenger RNA to make viral proteins? And then how do these viruses work to change a normal cell into a cancer cell? Do they carry genes? Do they do something to the host genome? And those seem like pretty profound questions that might have consequences for society if we could understand more about the origins of cancer. And those societal benefits that might result from work on these viruses, either focusing on the replication cycle of the viruses or on their ability to cause cancers, have indeed, as history has shown, led to effective therapies for a disease that wasn't even known at the time that many of us were moving from some other field to the study of these viruses to treat HIV/AIDS with drugs that inhibit the enzymes that are responsible for the conduct of the virus life cycle. And secondly, as I'll say a little bit more about in a moment, to targeted therapies for many cancers that have mutations that we understand as a consequence of having studied the oncogenic mechanisms by which RNA tumor viruses work. So I'm going to just tell you a little bit about some of the work that was done, especially to understand how these viruses cause cancer, just to remind you of how science proceeds and how the pace can be very slow at the beginning and very rapid once the right technologies are developed. So the study of retroviruses or RNA tumor viruses dates back to the early days of the 20th century, and especially to a moment in 1910 when a young physician at the Rockefeller Institute, Peyton Rouse, was given a chicken from a poultry farm on Long Island that was carrying a sarcoma, which is a solid tumor shown on the left. And from that tumor, Rouse was able to make an extract and pass the extract through a filter and to filter out animal cells and to filter out bacteria, and to find that there was a residual microscopic or submicroscopic component that we would now call a virus that was able to induce a sarcoma in an infected chicken. And the agent that caused that infection and that tumor is known now very widely as Rouse sarcoma virus, one of the first retroviruses and perhaps the one to be most intensively studied over the last 100 years or so. The second historical event of note is the reduction of this assay that I've just illustrated for you of the tumorigenic an assay for the tumorigenic capacity of this virus in an animal to cell culture, where it's much easier to do, much faster, taking cells from a chicken embryo, making a culture of a monolayer of fibroblastic cells, infecting with the Rous sarcoma virus, and showing that there were foci of transformed cells resulting from the infection. This meant that you could quantitate the number of infectious tumor transforming viruses in a culture and do quantitative virology, that you could begin to study viral mutants, including viral mutants that affected the gene responsible for the transforming activity of this virus, and you could begin to think about preparing large amounts of virus to isolate the genome and look at the proteins and try to figure out some of the aspects of the virus growth cycle. I don't have time to review, in the context of this kind of lecture, all the things that happen as a result of having this virus and a simple way to assay for it, but there are a few things that I want to stress about what our lab and several other labs who had chosen to pursue these viruses had learned, especially in the first decade or so after access to these viruses became feasible. The first is the recognition that virus multiplication depends on several viral enzymes for making DNA from RNA, reverse transcriptase, for integrating that DNA into the host genome, for expressing the viral proteins using cellular mechanisms and all of these factors that influence the replication of these viruses have become critical for confronting the epidemic of HIV/AIDS, for developing gene therapies, and for understanding the evolution of human genomes. Secondly, we learned from genetics that the virus carries at least one gene, that is, Rous sarcoma virus carries at least one gene that encodes an enzyme, a protein tyrosine kinase, that phosphorylates tyrosine in proteins, a novel kind of enzyme that proves to be fairly widespread and involved in lots of forms of cancer. And a special contribution that was made by me and Mike Bishop and our colleagues was that gene in the Rous sarcoma virus, a gene called VSARC, was derived from a normal cellular gene and functionally altered. That work was done by preparing molecular probes, radioactive cDNAs, that were specific for the region of the genome that it was shown genetically to carry the instructions for encoding the protein tyrosine kinase. And by using that radioactive cDNA as a specific probe, it was possible to show that normal cells had a gene that was nearly identical to the viral gene. We call that a proto-oncogene. It wasn't identical. There were several base differences that account for the fact that the cellular gene makes an enzyme with a very low level of constitutive activity, whereas the gene, the viral SARC gene that's been somehow captured from infected cells, carries a gene which encodes an enzyme that is highly active, and the high activity of that protein kinase is responsible for its transforming activity. But one of the most important features of this discovery, for which we've been more than amply rewarded, has been to recognize that this is not a phenomenon that is unique to Rous sarcoma virus. Indeed, as this iconic cartoon illustrates, there are many other retroviruses that are found in chickens, in mice, and rats, and non-human primates that encode other genes that are responsible for the oncogenic activity of those viruses that also derive from cellular genes. We now understand a great deal about how the capture of those genes occurs and how those genes are rendered cancer-causing when, under normal circumstances, proto-oncogenes serve important developmental and other functional characteristics in normal cells, but can become instruments of oncogenesis when they're mutated in certain ways. And particularly importantly, many of these genes have been found to be mutated in human cancers. And the result of understanding that and learning something about the biochemical and physiological properties of those genes has allowed the development of drugs that can inhibit the function of those genes when they operate in human cancers to propel the growth and other oncogenic features of those cancers, ranging from chronic myeloid leukemia, one of the first to be a target for the development of so-called targeted therapies directed against these oncogenes, in breast cancers and lung cancers and melanomas and several other cancers. And that has bred a whole story about precision medicine that I really won't have time to talk about. So that illustrates the principle of choosing a good question that leads to answers over many years and serves some societal benefit that encourages people who invest in science to keep investing. And I think it's important to keep in mind that sensing the potential for any scientific proposal to have the kinds of consequences that are illustrated here by the study of retroviruses is an axiom to live by in science. So the fourth axiom is a pretty simple one, and I think I've already indicated how important it's been to me that science is best practiced not simply as a solitary activity, but as a kind of team sport. And I've mentioned my long relationship with Mike Bishop, shown here with when we were running a Cold Spring Harbor meeting in 1978, when we were fortunate to receive the Nobel Prize in 1989, when as directors of what then became separate laboratories at UC San Francisco, kept our groups together by not just scientific meetings and collaborative work, but also by competitive sports, in this case, softball. And of course, our partnership was only the tip of the iceberg of a team, and one picture of a team activity is shown at the bottom left here. We're all celebrating together the good spirit that existed among a diverse collection of people: technicians, graduate students, medical students, postdocs, some wives, and husbands, all working together. And that relationship continues. We were Zooming together just last week, but this is the two of us teaching a class at Lindau in 2015. So, number 5. Here I wanted to step away from the simple conduct of scientific work to think about one other attribute of many jobs in science, which is teaching. I know a lot of people who say, well, when I get evaluated for tenure or evaluated for election to the academy, no one's going to care whether I was a teacher or not. They're going to care about whether I published great work in great journals and to great acclaim and won prizes for that work. But I would contend that teaching is really an essential feature of a life in science, and it's one that benefits not just students and institutions, but also the teacher, him or herself. For one thing, by exposing yourself to bright students, you're able to recruit the best students to your lab if you do a good job as a teacher. You'll learn things about your own field that you might not otherwise have learned, because you have to be the most knowledgeable person in the room. You do serve your institution and its trainees, and that public service is also satisfying. At least in medical school and graduate school teaching, it's very common to teach courses as part of a team, and you work with your colleagues in ways you might not have otherwise have done. And I think all of us learn even fundamental features of biology and chemistry and other fields by interacting with those colleagues during a teaching exercise. And then you can expand your intellectual boundaries quite widely by teaching, especially if you extend yourself as a teacher to areas in which you may perhaps not so familiar. And I'm going to spend a moment or two just talking about my own recent history doing that. So a few years ago, just after I returned to New York from being director of the National Cancer Institute, I decided that I wanted to do teaching of undergraduates, which I had never done before, and to give my own course, not simply be in a team course. And I wanted to extend the boundaries of my teaching beyond science itself to the question of how the scientific enterprise works and what it does for society. So I set up a course that has been taught for several years at the City University of New York and its Honors College. More recently, I've been teaching this at Columbia. This is a course about various aspects of science and how it relates to society. And it includes an effort to delve deeply into questions like why people choose to become scientists, why governments decide to pay for science to be done, who owns the products of science. This should be industry or NIH or the inventors themselves. What are the limits of science? What is wrong with the scientific process? How does science influence government policies of various kinds? How does science get portrayed in the arts? And this has been a fertile area for me, also for the students, but it illustrates how one's opportunity to influence the growth of young minds can also be a way to influence the continued development of older minds. So we're halfway through these axioms. We have a few more to go. I'm going to spend a couple of minutes talking about something that's been a passion in my life for the last 20 years or so, namely how we can make science more accessible to the public, something which I think is incumbent on all of us who are supported by public funds. It's clear the internet has changed how we approach this question, but even a long time ago, there were people who were concerned about this. So in the early part of the 17th century in Holland, a famous anatomist named Dr. Tulp assembled the citizens of Amsterdam and the surrounding areas to learn what he knew by sitting in on his dissection of a corpse, shown in this famous painting by Rembrandt. But about the waning days of the 20th century, I began to, as director of NIH, to think about whether NIH and NIH-supported investigators were doing enough to share our knowledge with the general public, which was increasingly interested in what medical science was offering to them and their families, and began asking myself whether we were treating our knowledge like ancient monks sitting in a small chamber with carefully illustrated documents, or whether we were behaving the way the New York Public Library would behave in opening its vast repertoire of books to the general public. Because I was in a position of some power as director of NIH, I was able to think about how we might expand our ability to disseminate knowledge. One obvious thing was to say, all right, people really don't have to go to the library and read a book or a journal on paper. We could spread knowledge much more rapidly by taking advantage of new digital tools and create a digital public library. Now, we were all familiar with PubMed, which delivered the names of authors and titles of papers and abstracts, but why not publish the whole paper online so that people can sit at home and they don't have to go to a big library full of medical journals to see what NIH and other publicly funded science had produced. And that led to the creation of a public library called PubMed Central that was able to format full text articles and make them accessible to anybody with an internet connection. And then we began to realize that there was a potential to make journals as accessible as digital libraries by encouraging the journals that were already beginning to publish their subscription-based journals online to create journals that were online journals that had a business model in which the authors or the funders of the research would pay the actual costs of reviewing and publication, and then make the final papers available to anybody who had an internet connection without a subscription base. And that had a number of advantages compared to subscription-based journals, at least in my view. Subscription-based journals having high subscription fees that were making huge amounts of profit for the publishing companies, taking advantage of free labor provided by the authors of those articles, the reviewers, the editors. This was a business plan that was too good to be true, but was true, and was gathering billions of dollars for big publishing houses like Elsevier for many years. And the publisher would control the copyright, so the autonomy of scientists was limited by that. And the contents were not in public libraries unless there was some mandate for, or some unusual individual who would sponsor the deposition of work into the public library like PubMed Central. Where in contrast, open access journals like the one that some colleagues and I founded in 2003, the Public Library of Science, were free to anybody who had an internet connection, and with the necessary small fees paid by from the grantees, paid by the granting agencies or by the authors who were paid by the agencies, with the authors retaining copyright and all contents deposited in libraries like PubMed Central. Now, this is a controversy which has not yet fully abated, but there are lots of options for open access publishing now. Moreover, there are other ways to get information into the public domain even more quickly. One of those is to make use of what are called preprint servers, that is digital databases that contain work that has not yet been subjected to peer review, but has been contributed by authors who are ready to show and share their work. The most prominent example in our field now is bioRxiv. And let me just say a word or two about the preprint movement, because biomedical science has come late to this game, but it is serving an incredibly important function now. So we come late to the game because physicists, computational scientists, mathematicians, and astrophysicists have been posting their articles before review and official publication since 1991, when Paul Ginsberg, who's a colleague of mine at Cornell University, developed something called Archives. Archive, for very few dollars, posts work that then is subjected to review by informal review by colleagues, and later those articles are submitted to formal publication. But the work had been out there from the moment that the authors felt they were ready to share their work with their colleagues, and essentially get gossip back in return, postings of comments on the internet. And it wasn't until a few years ago that Ron Vale, a colleague of mine who will be talking in Bangalore, I believe, tomorrow or the day after, decided to push the concept of preprints, which I and others had advocated, but without a lot of success some years earlier. And the idea that the advantages of doing that were probably clear to most forward thinkers, but Ron sketched them out and wrote about them, and developed an advocacy organization called ASAP, As Soon as Possible Bio, in 2015. And now it's been everything that I've been talking about in the way of making science a more open process has developed pretty slowly, and that's developed slowly in part because scientists, at least American scientists, are inherently quite conservative. They love their traditional journals. They're very respectful, too respectful in my view, of the prestige of certain journals, especially Nature, Science, and Cell, that have very high journal impact factors, and publication in those journals has become a common metric for evaluation of individuals, for jobs, for tenure, for promotions, for prizes, and that has made people feel constrained about changing the way we do science publication. The commercial publishers have pushed back on this movement because they enjoy those large profit margins. Many scientific societies, to my great disappointment, are loath to abandon the traditional means of publishing through subscription-based journals because they use the profits from those journals to support many other activities, valuable activities, but at the price of avoiding doing what they ought to be doing, which is to make their scientific results more readily available. But there have been countervailing forces as well, and that's been quite important, that there's been commitment of faculty to make changes in the way we distribute information, illustrated by a long fight between faculty at the University of California against the efforts of Elsevier to keep prices for journals at a high level. By the growth of the preprint servers I've talked about, and of course during the pandemic, as I'm sure you're all aware, preprint servers have become an indispensable means to get results into the public domain quickly at a time of public health emergency. Then some political forces in the U.S. have worked effectively either through Congress, which has demanded deposition into PubMed Central within a year after publication in 2008, and more recently, the Biden White House has said that things must be deposited immediately without embargo into PubMed Central through some form of open access publication. And there are movements that come largely from Europe to mandate open access publication through something called Plan S. So that's it's unfinished business. We can come back to it in the discussion. And I'm going to move on to Axiom 7, which has to do with the relationship between science and society. I think we all recognize, if we've received money from public funders, such as governments, that science depends on support of society through such funding mechanisms, through patient advocacy, through advocacy for science itself, and that scientists ought to think about how they're going to serve the scientific community that is supported in a public way by society. My own history at UCSF, University of California, San Francisco, illustrates the way in which people may change in their approach to this kind of service. So for the first 10 or 15 years that I was on the faculty there, I admit that I was an obsessional lab scientist focused on my own research on retroviruses and cancer genes. And I really didn't participate in the society of my own institution. I never went to a faculty senate meeting. I never wanted to be a departmental chair. I didn't join committees unless they were involved in the selection and the nurturing of students. But as I grew up, as I became more confident as a scientist, my attitude changed. I was involved in international committees, such as the one that named the AIDS virus HIV. I became involved in supporting efforts to provide more funding for AIDS research. I got engaged in disputes about misconduct in science and in the way in which the government supported or failed to support the indirect costs of doing research. I was an advocate for funding of young investigators. For senior scientists at UCSF, my colleagues Bruce Alberts, Mark Kirshner, and Mike Bishop got together with me to write a letter to the newly elected president and vice president, Clinton and Al Gore, to say, here are the things that are wrong with NIH that you can fix. Among those are increasing the budget so that it will double over 5 years, and many other things that too numerous to discuss here. The result of all that was that I suddenly found myself as a candidate for being the director of this agency, and went from an ability to give some advice as a senior member of the scientific community to being asked to take some action to make these things actually happen. So in November 1993, I became the director there and found that that public service of serving as director of NIH had a lot of value and was associated with a lot of good times. Fortunately, I was working at a time in which the administration was supportive of expanding scientific activity. The NIH budget was not only growing, but finally Congress and the administration adopted the view that doubling the budget over 5 years was in fact a good idea. And because the environment for doing science was improving, it was a good time for recruiting new people to the NIH, as Partha indicated in his opening remarks, for expanding certain things we were doing, building new buildings, and most importantly, contributing to a lot of pretty critical advances in genomics, where the Human Genome Project rolled along at an increased pace and was semi-completed by the time I left NIH. Our work on global health was expanded. As you've heard, we developed PubMed Central, a digital science library, and the work that was being done in cancer research led to the development of targeted drugs, and work that had been done by the retrovirus community had allowed the development of effective multi-drug treatment for AIDS. We created a new center called the Vaccine Research Center that's been whose value has been proved during the pandemic, because it's the place where some of the technology essential to RNA-based vaccines was developed. We built a new clinical research center and developed clinical research training programs that have been influential everywhere in the world. It was also a good bike route to work, but that's beyond the concerns of most of you. To illustrate some good moments in this period of public service, I show a picture of the most famous scientist in the world, Tony Fauci, at the board, talking about the details of AIDS virus replication to president and vice president. The guy with the bald spot is me, down the lower left. And the picture on the right shows one of the more famous women in the world, Hillary Clinton, cheering along with me at the time of the 1998 State of the Union address by President Clinton, when he was announcing his intention to dramatically increase the NIH budget. So this part was fun. There were some parts that weren't so much fun, but these were memorable. So I want to reflect a little further and a little more deeply about the relationship of science to society through politics, with a couple of historical reminders that science has been supported in our country because of our political history, and science will never be free of politics if it's conducted in a public fashion. So we are a country that is a product of the 18th century Enlightenment. I don't have time to get into that in great detail, but it's illustrated in some ways by one of the great years in Abraham Lincoln's tenure as president, 1863, in which, in addition to conducting a war against the rebellious southern states, he did three things that were incredibly important to science. One was founding the National Academy of Sciences. Secondly, finishing a science-based project to build a railroad across America, and he passed legislation that established so-called land-grant colleges, where most agricultural science in our country is conducted. During the Second World War, the contributions of science to the victory of the Allies, through the generation of atomic bombs, through development of therapies against a variety of diseases, through the development of sophisticated radar detection, led Franklin Roosevelt to say, we ought to have a policy by which the government oversees more science than it currently has done. And he asked the man who was in effect his science advisor at the time, Vannevar Bush, to write a prescription for how the U.S. government should oversee science. And Bush wrote a very famous tract, which I'm happy to be teaching every year to undergraduates, Science, the Endless Frontier, that outlines a blueprint for how science should be done in America, with the federal government sponsoring fundamental research at universities through grant programs, with the knowledge generated by that research being taken advantage of by the industrial, commercial communities to make things that are applicable to the solution of practical problems in the private sector. Better things for running a domestic household, better cars, better drugs, better everything. President Nixon in 1971, by engineering and then signing the National Cancer Act, dramatically increased the amount of research being done on cancer and how the NIH was going to be structured. All presidents, with the exception of Mr. Nixon, had councils of advisors on science and technology. I happen to be co-chair of the Obama PCAST in the early parts of his term. And as you can see from this assembly, Obama and many other presidents have sat down with scientists to try to take advantage of their knowledge to combat a variety of threats of all kinds, as illustrated by this discussion that we're having about the influenza vaccines that were important during the influenza pandemic of 2009, 2010. And President Obama himself has been engaged in development of new initiatives like the Precision Medicine Initiative that he was announcing when this picture was taken in 1910. Briefly, something about the essence of scientific work and its relationship to the world. I've been a strong proponent probably ever since my visit as a medical student to India in 1966. I hate to divulge that number, but there it is. I've learned that science is inherently an international activity, and that we have to be careful, especially in these times of growing nationalistic policies, felt very dramatically during the Trump administration by many of us. We can't let Brexit or other right-wing leaders spoil the international characteristics of science. And you can view that international flavor of science either by looking at individual labs. This is my lab group when I was at Memorial Sloan Kettering, with the countries of origins listed here. You can see virtually everybody comes from some country other than the USA. That's been true in my lab for many years. But it also extends to the way in which international organizations work together. For example, I teamed up with the head of the Pasteur Institute when I was NIH director to start an organization called the Multilateral Initiative on Malaria, by bringing people from various countries together to meet in Dakar, Senegal, to establish this new organization, which was particularly focused on developing good malarial science, conducted by Africans who would learn to be good scientists, and were willing to collaborate to foster the development of malarial science across Africa. But the U.S. has funded a number of efforts to internationalize science. The Uganda Cancer Institute in the upper left, the PEPFAR program, which is mainly a care and prevention program, but also has research components. It's been involved in malaria research for a long time in Bamako, Mali, at the Malaria Research and Training Center. And those are activities that we don't want to lose in this burst of nationalism that is troubling me and many others about the conduct of all things, but especially science. This is a list of some of the things that worry me in America at the moment. The strong Republican voice and for America First based policies, some immigration procedures that are discouraging people from coming abroad, the sense of competitiveness among nations who are conducting science, which should be an international activity, and the ethnocentric paranoia, especially against China, that is currently rampant in the US government. Finally, one very short item which we don't want to lose sight of, and that is that science is more than just an effort to deliver new ideas and make new products and create a more comfortable life for everybody. It's also a source of incredible inspiration about questions that go a lot deeper than how can we improve health, but have to address themselves to the question of what the universe is and why it is here and why we're here, and whether it's looking at kilonova through remarkable telescopes and LIGO detection devices, whether it's discovering the rules of life by learning about how genetic instructions are encoded in cells. These are things that should inspire all of us to think that science has the potential to at least partially explain what it means to be human and what it means to be alive and why the universe exists in the first place. So if I tried to say anything here in this rambling talk about the axioms by which I've lived, it's to say that a career in science is a remarkable privilege, and a privilege that doesn't come to everybody. That can be a source of distress, but it's also often the source of pleasure and gratification. But it also comes not just with benefits to society, but with costs to science and to society, and responsibilities that all of us need to recognize and accept and pursue. Thank you for listening. Sure. Oh, absolutely. If it's not too late.
Are there any questions for Dr. Varmus? Speak up or, yeah.
Let's move the microphone around. Yeah.
Hello. So, do you think like science should be time bound? Because in at least in our institute, so science is like PhD course, like work is bound in 5 years paid. Then after that, it is on your own. So, do you think that should be changed at the administrative level, that science should not be time bound, or if it is not finished within 5 years, not for due to the student's fault, it should be like compensated by the administration?
Yeah. Well, I think what you're saying, I don't want to get too deeply involved in local politics because I feel there is an agenda here. But I think we have to acknowledge certain things. That at one level, the time it takes to do a scientific project is very difficult to predict. Some projects are done in a few weeks, some are done in a few years, some are never completed, some take years. We have to support scientists with grants, with salaries, with health insurance policies. We need to do a certain number of things that are done under contract with certain rules. And one of the things that we face in our country, to give you one sense of the time problem, is that Congress likes to assert itself every year by providing a budget for agencies like NIH or the National Science Foundation. Those agencies, the NSF and the NIH and others, recognize that almost no science is done in 1 year, so they give grants or contracts that last 3, 4, 5, sometimes even 7 years. So that creates an immediate tension because the agencies don't know how much money they're going to get in year 2 when they're giving away in year 1 commitments to support research for several years more. So the same thing happens when your institution gives you a contract to work for 5 years and you take a project on that might be done in 3 years, 5 years, or 7 years after you're gone. So I just think there are realities to the time here that involve the uncertainty of science and the need to have some certainty in the way in which your science and your salary are going to be funded.
Harold, I understood from your talk that right choice and right time is important for success in life. And somebody's choice may not be good or not good for someone else. So the question to you is, both RNA and DNA tumor viruses, they cause tumor, right? So you worked on RNA tumor viruses. The work on RNA tumor viruses has started much earlier than DNA tumor viruses, if I'm correct? No?
No, In the early 1960s, some remarkable advances were made in the study of DNA tumor viruses that long preceded our understanding of how RNA tumor viruses work. What I and many others found particularly attractive about RNA tumor viruses is that you could separate the replication functions, that is the functions devoted to the multiplication of viruses, from the oncogenic functions. With the DNA viruses, the genes that promote tumor formation are the same genes that are required for replication of the virus. That to me smelled like trouble. And the idea that you could separate replication functions from transforming functions proved to be incredibly important in
Yes,
making
I agree.
progress.
RNA tumor viruses, actually studying them led to the identification of cellular oncogenes, while DNA tumor viruses identified and also established functions for very important proteins like P53, RB, P300, the recent
I
time.
have to say I'm going to argue with you about that.
Okay.
I mean, it would have been nice if that actually had happened that way, because you're right that those viruses do depend on the ability of certain genes, like their early replication genes, to suppress the activity of tumor suppressors. However, I would make the argument, and I have a lot of evidence to support me, that a gene like RB was not discovered that way. It was discovered by recognizing there was a disease called retinoblastoma, and that this was caused by a recessive genetic phenomenon, and the gene was cloned, and only later did people like Harlow and Livingston and others show that one of the ways in which the large T antigen worked was by interfering with the function of the RB gene. But the RB gene was not discovered as it should have been.
The later discoveries are no less, Herald, because the gene was discovered, the protein and the functions were established by studying DNA tumor viruses.
Yeah, later.
So the question is, I will stop here.
Yeah, I'm not saying that they're totally separate, but I just think that...
The question to you, I'll just make it brief, only
okay.
10 seconds. Instead of choosing RNA tumor viruses, you had chosen DNA tumor virus to work with. Your major discoveries came in late 70s. So what would have been your path, and what kind of discoveries you would have made?
I don't know. That's a cocktail party discussion. I think about that sometimes, but I'm glad I went the way I did.
Thank you for the talk, sir. I am a PhD student in neuroscience, and I have two questions which are interrelated. So, has science changed from your time? Because when I get,
I think this is still my time.
Yeah, I will rephrase it from 1970s or so. So,
well, that was my time too.
like whenever I get a groundbreaking project idea, I always end up finding that it has already been pursued. Sometimes even as back as 1950s.
Because you
or
chose the wrong
1960s.
question. You've got to read the literature first.
So, like the amount of literature that has accumulated in past decades has increased so much that is the quest for a good idea becoming increasingly difficult? That's my question.
That's an interesting question. I don't know if it's more difficult or not. One of the developments that's very striking to me is the idea that less and less research is done in a hypothesis-driven fashion, that more is being done by walking along the beach and looking for unusual seashells. That is, using some powerful genomics tests, screening for organisms with unusual properties, or asking whether there are cancer cells that are being driven by some unexpected activity, using an RNA array or something like that, and then trying to develop a question after you get a lot of data. So I think there's a new way of doing science that begins not with the question itself, but by using a powerful technique to gather a lot of information and seeing whether any of it is provocative. So one of the things that I try to do in my own lab is to survey, for example, information about cancer genomes and look for things that are unusual. About 10 or 15 years ago, people began to realize that although lung adenocarcinomas are likely to have an EGF receptor mutation. That's found in about 15 or 20% of those tumors. And it's likely to have a mutation in a RAS gene, maybe 35% of tumors. But no tumors have both mutations. Why is that? So then, we had some ideas about why there is this mutual exclusivity. Why is that? It turns out that putting both of those genes into a cell at the same time is toxic. So that's interesting. Why is it toxic? Well, it turns out to be toxic because you get a very high level of activity of a gene in a protein in the signaling pathway called ERK2. What is ERK2 doing? Is there some way you could make ERK2 particularly excited in a cell that already has a cancer phenotype and has a mutant RAS gene? And could you use that as an approach to cancer therapy? So that's a different way to think about problems than was true in the past. So I think the way in which we come to the best questions has been changed, maybe enhanced by the fact that we can survey with these new powerful technologies so many attributes of cells without asking a question, just seeing whether anything usual pops out that we need to understand. But I don't think there's any shortage of good ideas. I think you just have to be pretty difficult on yourself in thinking about which questions are really worth addressing. And you certainly want to start by being sure that nobody else has answered them first.
Hi, since we are talking about careers, do you wish to, or were you ever offered to be the chief medical advisor to the president? And in that context, okay, in what capacity does the chief medical advisor help in?
Well, there really is no chief medical advisor in the U.S. government. There's a chief scientific advisor. And as you may know, traditionally, the medical advisor or the scientific advisor to the president has generally been a physicist, because historically, the big thing that presidents are worried about in the scientific domain is the military power of other countries, weapons, and the potential for something that could be destructive. It's only in the last, say, 5 or 10 years that people have given more thought to the importance of having, as a White House science advisor, somebody who is focused in biology. There are obviously a lot of other ways in which biologists can serve the government, being director of NIH, being director of the CDC, the Center for Disease Control, being the head of the Food and Drug Administration, working at the National Science Foundation. So it was only in the Biden administration that a biologist, Eric Lander, was chosen to be the science advisor. I think that will happen again. I think it was a healthy development, and the pandemic certainly led people to recognize that having a science advisor who is medically educated is good. There has been, in the person of Tony Fauci, a chief medical advisor during Dr. Fauci's last days in government. He was the chief medical advisor, a new position to Joe Biden during the pandemic.
Hello. Yeah. Thank you so much. It's more of a question/comment. You talked about open access to scholarly knowledge and scientific knowledge. IISc being one of the top scientific producers of the country, both in terms of quantity and quality. We do have institutional repositories, national repositories, and all of those. But the amount of papers and scientific output that is populated there is less than 1%. And it's been there for the last 3-4 decades like this. As someone standing in front of us, to the scientific community, both in IISc and in India, what would be your message in terms of importance of open access?
Well, my view is that all scientists have a responsibility to get their work into the public domain as soon as possible. I am very uncomfortable as a recipient of NIH funding to say to librarians, to disease advocacy groups, to school teachers, to journalists, that you can only see the result of my work if you pay a subscription fee to journals to read those papers. Now, I know, I was waiting for a question, because I've had questions asked to me earlier in my trip, how do people who are doing work with limited budgets in countries that are not wealthy pay the the so-called author's fees for publication. And I think it varies a lot, but I think we should keep our eye on the goal of getting the information into the public domain. So in some cases, it may mean that you don't publish your work in an open access journal published by Cell, which will charge you $7,500 to publish an open access paper. But when mandates to put your work in the public domain come from governments and funders, we're going to figure out cheaper ways to do things, and that's already happened. So you can do that in various ways. One is to say, I will put my work into the public domain as a preprint before it's even been reviewed by a journal. And it's not going to change very much in the review process. It may be improved slightly, but the fundamental properties of what I've done are there. The second thing is to reach an agreement with the journals that you're not going to pay them these high prices, but that you're going to post onto an appropriate digital library or some kind of server a version of the article before it's been edited and formatted by the journal, and it won't look as pretty as it does in the high cost, high prestige journal, but the paper will be there. It's the accepted form of the paper, and it just doesn't look as pretty. Another thing to do is to say, I'm not going to post my paper in a digital library. I'm going to post it on my own website and figure some way for people who want to read my paper and see the title of it to go to my own website. That's a cumbersome solution, but it's a partial solution. Now there is a new development in publishing that I think is very important, and that is the publication of reviews. Those reviews can be on preprint servers. They can be published along with a paper when it appears in an open access or a subscription-based journal. That takes some of the pressure off the need to spend months revising a paper. The author has the right to make the decision when to publish his or her work, and to include at the time of publication or the time of dissemination accounts by other experts who say the following should be done. There's a very troubling tendency these days for people, especially reviewers, to want a story to be absolutely complete. Well, as I've tried to indicate, good results prompt new questions. Things are never done. But what you want to do is keep up the pace of delivery of results as they come through with criticisms that may indicate that the following questions remain unresolved in this work. But the pace of research, as the pandemic has shown so clearly, should be higher than it is. People are getting a lot of data, but the time between publications, in my view, has gotten too long. And that's in part because certain journals that exert great influences over the development of scientific careers are placing unreasonable demands on authors before their paper appears in those journals. And we have to counter that, and we have to keep in mind that we have a moral imperative to deliver to the people who support us the answers that we're getting, and they shouldn't be hidden behind subscription barriers.
Hi. So, have studied humanities helped you think in science?
Yeah. Where are you?
I'm
Oh, yeah, okay, yeah.
like, have humanities helped you think in science, to do science?
I don't know. Maybe, maybe not. Certainly studying, as I did, studying literature for a long time, probably made me a better writer. You don't have to be a scientist for very long to learn that being able to write a good grant application, to write a good paper is pretty damn important. But, you know, I think that studying, whether it's history or the arts, is just, you know, something that's valuable to a good life. Doesn't need to be something that promotes careers.
Sir, as viruses just want to forward the information contained in them, is it likely to...
Where are you?
Yes.
Okay. Yeah.
Is it same with all the other organisms?
I don't hear the question.
Sir, as the viruses just want to forward the information content in them, is it same for all the other organisms? Or in other words, can we say that we are just some information being forwarded by some organic molecules?
Gosh, I don't think I really understand the question. I mean, studying viruses is studying viruses. Viruses are, first of all, they're very diverse in their own attributes with different kinds of genomes, different routes of replication. They teach us a lot about the rules under which biology act, but all classes of viruses have their distinct properties. They're often useful for helping us understand that many properties of cells have been first understood by studying viruses. And a good example is reverse transcription. That was thought to be, people didn't believe Howard Temin when he argued that retroviruses operated through a DNA intermediate because there was no enzyme. Well, once the enzyme was discovered, then it turned out that telomeres, the ends of our chromosomes, are generated by reverse transcription, that there are movable elements in human cells that move around as a result of reverse transcription, that hepatitis B viruses have a reverse transcriptase that is an essential feature of its life cycle. So they're transposable elements in yeast that depend on reverse transcription. So we often learn some fundamental principles of biology by studying atypical organisms like viruses, but viruses don't violate rules of biology. They sometimes establish new rules. So I'm not sure whether I'm answering your question, but viruses are unusual because they sometimes display properties that would not have been appreciated without actually looking at the viruses themselves. I think a useful thing to think about is how prions work. How does a protein-based element that is, in a sense, infectious operate without an apparent genome, without RNA or DNA? And that's something we're thinking about. I don't think it violates rules because it does happen, and the rules have been expanded. Think back to the first articulation of the central dogma by Crick, who said that information's in DNA is expressed by making RNA, and RNA is translated into protein. People interpreted that in the wrong way. They thought that it meant that information could never flow from RNA to DNA, and he didn't say that. So sometimes we don't appreciate the full implications of what seems to be a principle because the understanding of what the rule is not properly interpreted.
6:30, one last question.
Hello, sir. Thank you for your wonderful talk. So, a year before, I had an opportunity to present one of your seminal papers where you established that this SRC gene is in itself is in human. So,
sorry, I don't know. You spoke too quickly. I didn't understand what you said.
Okay. So, I had a question regarding that when I presented your paper. So, in that paper, you have established that.
So, bring that microphone closer.
What is the paper?
So, he presented one of your papers in a journal.
Okay. What was the paper?
Where you have established the fact.
Cellular origin of the VS.
Okay.
Yeah,
all right.
So, my question is like, what is the origin of SRC gene? Like, is it viral or is it humans? During the evolution, it has jumped from the virus to humans.
No, the Sark proto-oncogene is a cellular gene. It was acquired, and it gets acquired repeatedly by viruses because viruses have the potential. The way we think this works is that a virus without an oncogene, a virus that just has the replication functions, making the core proteins, the enzymes, the envelope proteins, and the genome, has the potential to infect millions of cells in an organism. And one of the properties of the retroviruses is they make a DNA copy of the genome, which gets inserted more or less randomly into the host genome. One of the things that happens during the process of semi-random integration is that some genes are inactivated by those insertions, some genes are turned on. sometimes turning on those genes, like the WNT1 gene, the MYC gene, several other genes, can lead to a tumor in those animals. Then the cell that has the mutated, insertionally mutated gene expands because it has enhanced growth properties. Then you have millions of copies of those cells. Retroviruses have the potential to undergo genomic recombination. And just rarely, there may be recombination between the viral RNA genome and the RNA from the cellular proto-oncogene. And when that happens, you generate this cancer-causing virus. That virus usually doesn't replicate well at all, and it will disappear unless the animal is taken to a scientist who's interested in a virus that might be a cancer virus. That's what happened with Peyton Rouse. Some poultry farmer brought him this chicken with a tumor, and then he isolated probably a virus with pretty low, what we call titer, probably very small amounts of virus, but he wanted the virus that causes the cancer. So he isolated that virus, and then he studied it. And so it seems as though it's widely used, but these events are very rare. And they come about because of the unique replication properties of retroviruses and their ability to acquire cellular information. And when you think about that, you say, that's gene therapy, that's gene vectoring. That's using a virus as a vector to carry a gene. And everybody who does modern molecular biology uses genes as vectors, simulating what happens at very low frequency in nature when these viruses cause tumors.
So another question came in my mind when I was reading your paper. It was a tough deal. So I read 5 to 10 times that paper. So my question is...
I thought that paper was easy to read.
Yeah, I don't think so. So my question is, why do people write...
So it wasn't clearly written?
Why do you write in such a complex language? Like, it's very easy. It's very tough.
I thought I was a good writer. Maybe it's Mike's fault.
2 more questions?
Sure, it's up to you.
Okay.
Yeah, I'm fine.
One here, and we end there.
I'd rather answer questions than give a talk anytime.
Hi, sir. It was an excellent talk, and I have learned a lot about from your lecture. But something I don't know is it a relevant platform to ask or not?
Sorry.
Yeah. Please accept my apologies if the question is irrelevant here. The question I would like to ask: everyone has a success stories, right? They might be having the hardships as well. May I know what are the hardships of you have faced when you would like to introduce a lot of digital platforms, or you have been introduced many, so you might be had very hardships to encounter to actually make it functional. I would like to hear about it more.
Yeah, there are 2 kinds of disappointment, maybe 3 kinds of disappointments I've had in science. One is letting someone else get the answer before I do, and that's happened a few times. I've just been scooped. We're both pursuing the same question. I chose the wrong route. Someone else got there first. That's pretty trivial, but it does happen, and it's disappointing, or it's at least aggravating. The second thing would be making a bad choice about a person I brought into my lab. Lab communities are very intense and very intimate, and if you bring a person into your laboratory who turns out to be difficult to work with, not socially adept, not generous, that can really poison the atmosphere of a lab. So that's happened to me a few times, and those have been very painful moments. Third thing is when I choose to do a project that's not ready to be pursued. And I've occasionally even encouraged some of my best trainees to try to solve a problem that ought to be solved, but not yet, because technologies just are not there. So it's painful to watch somebody really talented working on a problem for which the techniques are not yet available. And that's probably the most frequent source of unhappiness running a research laboratory.
How did you handle this?
What?
Hi.
Hi.
Very briefly, I just wanted to ask you that I think you sort of alluded to this in one of the answers, but like eLife has this new way of publishing where they say you submit your work and then they will publish the peer review comments separately and then it's up to the reader. So I was just
Well,
wondering.
it's more than that. Let me just point out what I think is the central feature here. What eLife is doing under the direction of my good friend and co-founder of Public Library of Science, Mike Eisen, is to say we want to give back to the author the right to say when he's going to publish. When Galileo was doing science, he decided when he was going to publish his book. Nobody decided for him. And in the current environment, most of us are dependent on some editor or some reviewer to say whether or not our paper can be published. We should have the power to decide when we want to tell the community what we're doing, because we're the ones who are really at risk of being told that we're wrong. You don't want to be wrong. So what Eisen is saying is that if we look at the paper briefly, we decide it's worth reviewing, no matter how negative the reviews are, the author can choose either to respond to the comments or just go ahead and publish his paper with the reviews published next to him. So I think that's a really good way for people to make decisions about whether they think the critiques are fair, whether they're ready to go public with the paper. And people are doing that anyway now. I mean, my lab puts every paper that we are interested in publishing onto the bioRxiv preprint server anyway. So it's in the public domain regardless of whether Nature decides they want to officially publish it. So we're already taking a risk about whether people think that it's any good. So go on to your question. I'm sorry.
Yeah, the question was like, how do you sort of reconcile the need for like your trainees as well as your lab to publish in a journal with high impact factor that helps them set up their future as well as you get further grants, at the same time for your need to like do what you think is right, which is like open access and publishing when you think and not like...
Yeah, that's a really good question. It's one that people who come to my lab know that they're expected to publish, but both post their papers as preprints and to publish in open access journals. That's just the way we operate. But you know, I've had a few trainees who, after coming to the lab, say, I must publish in these high impact journals. And you know, I usually let them try it. Often they fail because these journals are incredibly selective, and then they end up publishing in a slightly less prestigious journal, but the paper gets out there, and if it's valuable, people respect it. And the last person to be of that phenotype was someone who finally got her paper published in the Journal of Experimental Medicine, and she got a very good job at a prestigious university, and it didn't hurt her career at all. So I think people sometimes inflate this idea. There is a reality here that unfortunately many search committees begin the search by looking through the applications and only interview people who have multiple papers in so-called high impact journals. I think if you have a chance to hear Ron Vail when he comes to town, he'll probably talk about a new adventure he's undertaking at Howard Hughes to try to get rid of this dependence on high impact factor journals. Get people to read the papers that people have written to see whether the work they're doing is exciting and well presented. We're embarrassing ourselves by depending on these high impact factor criteria. I don't know anybody who works in a Department of English or a Department of History who would make that kind of assessment of a job candidate. They would read the work. And often these are not articles, they're books. And if we want to have intellectual integrity, we have to do some reading. Now, we can't read 200 papers. So one of the things that all of us seem to favor, at least I favor, and I think Ron does too, is the idea that the applicant for a job or a prize or anything else should be singling out the most important work and saying in a paragraph why the work's important, what contribution he or she made, and then have the reviewers, the evaluators, read that work and see whether the assessment that's made by the candidate is fair.
Thank you, Dr. Varmus. I think we should give him a big hand one more time.
Oh, that's mine too. Yeah, I want that. Thank you. I paid for this. Okay, thanks a lot.
Leave your wants to do you some honors.
Oh, sorry.